Welcome back, everyone. Myself, Khalid Umar from Davao Medical School Foundation, first year MD program, Philippines. This is a continuation video on sensory physiology of taste and smell. So, if you haven't watched the previous video, please go and watch it. So, before we move on to the objective questions in detail, let's have a glance on the observation and result of the experiment. So as we can see in the observation table, we have got the two containers A and B. The container A contains a sweet dish and container B contains a biscuit which is salty. So the patient has identified the container A food items as a sweet, having a too sweet taste, and container B item having salty taste. So from this we can conclude the patient has got a normal sense of taste and smell as he has identified the taste of both the food items correctly. Coming to the objective questions. The first question is on the mechanism of chemoreception. So, chemoreception is a process by which organisms respond to chemical stimuli in their environment that depends primarily on the sense of taste and smell. Chemoreception relies on chemicals that act as signals to regulate cell function, without the chemical necessarily being taken into the cell for metabolic purposes. The second question is on the mechanism of olfaction. So the olfactory neurons project from the olfactory epithelium to the olfactory bulb as a thin unmyelinated axons. The olfactory bulb is composed of neural clusters called glomeruli, and each glomerulus receives signals from one type of olfactory receptor. So each glomerulus is specific to one odorant. From glomeruli, olfactory signals travel directly to the olfactory cortex and then to the frontal cortex and the thalamus. Recall that this is a different path from most other sensory information, which is sent directly to the thalamus before ending up in the cortex. Olfactory signals also travel directly to the amygdala, thereafter reaching the hypothalamus, thalamus, and frontal cortex. The last structure that olfactory signals directly travel to is a cortical center in the temporal lobe, important in spatial, autobiographical, declarative, and episodic memories. Olfaction is finally processed by areas of the brain that deal with memory, emotions, reproduction, and thought. Now, coming to the third question, which is on gustation. The taste neurons project from taste cells in the tongue, esophagus, and palate to the medulla in the brainstem. From the medulla, taste signals travel to the thalamus and then to the primary gustatory cortex. Information from different regions of the tongue is segregated in the medulla, thalamus, and cortex. In humans, there are five primary tastes, and each taste has only one corresponding type of receptor. Thus, like olfaction, each receptor is specific to its stimulus, or you can call it a testant. The transduction of the five tastes happen through different mechanisms that reflect the molecular composition of the testant. A salty testant containing NaCl provide the sodium ions that enter the taste neurons and excite them directly. The sour testants are acids and belong to the thermoreceptor protein family. Binding of an acid or other sour tasting molecules trigger a change in the ion channels, and these increase hydrogen ion concentration in the taste neurons, thus depolarizing them. The sweet, bitter, and umami testins require a G protein coupled receptor. These testins bind to their respective receptors, thereby exciting the specialized neurons associated with them. Before we end this session, let's have a look at few exercise questions to help you understand the sensory physiology of gustation and olfaction better. So the first exercise question is, what is the role of the olfactory mucosa when stimulated during normal respiration? The answer is, olfactory mucosa carries a specialized sensory organ for modality of smell. When olfactory mucosa is stimulated during normal respiration, the olfactory receptor in mucosa produce a generator potential as it binds certain molecules inhaled from environment by sending impulses directly to the olfactory bulb of the brain. The second exercise question is to describe how taste and smell sensation are created and interpreted. The answer to the question is, odorants or the odor molecules enter the nose and dissolve in the olfactory epithelium, the mucosa at the back of the nasal cavity, as illustrated in the figure. The olfactory epithelium is a collection of specialized olfactory receptors in the back of the nasal cavity that spans an area about 5 centimeter square in human. Recall that sensory cells are neurons. An olfactory receptor, which is a dendrite of a specialized neuron, responds when it binds certain molecules inhaled from the environment by sending impulses directly to the olfactory bulb of the brain. Olfactory neurons are bipolar neurons, neurons with two processes from the cell body. Each neuron has a single dendrite buried in the olfactory epithelium, and extending from these dendrites are 5 to 20 receptor-laden, hair-like cilia that trap odorant molecules. The sensory receptors on cilia are proteins, and it is variation in their amino acid chains that make the receptor sensitive to different odorants. Each olfactory sensory neuron has only one type of receptor on its cilia, and the receptors are specialized to detect specific odorants, so the bipolar neurons themselves are specialized. When an odorant binds with a receptor that recognizes it, the sensory neuron associated with the receptor is stimulated. Olfactory stimulation is the only sensory information that directly reaches the cerebral cortex, whereas other sensations are relayed through the thalamus. The figure shows the human olfactory system. It has got bipolar olfactory neurons extended from the olfactory epithelium, where olfactory receptors are located to the olfactory bulb. So we have talked about the smell mechanisms and the interpretation. Now coming to the taste. Detecting a taste is fairly similar to detecting an odor, given that both taste and smell rely on chemical receptors being stimulated by certain molecules. The primary organ of taste is the taste bud. A taste bud is a cluster of gustatory receptors, taste cells, that are located within the bumps on the tongue called papillae, as you can see in the figure. There are several structurally distinct papillae, filiform papillae, which are located across the tongue. They are tactile, providing friction that helps the tongue move substance, and contains no taste cells. In contrast, fungiform papillae, which are located mainly on the anterior two-thirds of the tongue, each contain 1 to 8 taste buds and also have receptors for pressure and temperature. The large circumvallate papillae contain up to 100 taste buds and form a V near the posterior margin of the tongue. The figure shows the first a foliate, circumvallate, and fungiform papillae, which are located on different regions of the tongue. And the second figure is on the foliate papillae. There are prominent protrusions on the light micrograph. Now, in addition to those two types of chemically and mechanically sensitive papillae are foliate papillae, leaf-like papillae located in parallel folds along the edges and towards the back of the tongue, as you can see from the figure. Foliate papillae contains about 1,300 taste buds within their folds. Finally, there are circumvallate papillae, which are wall-like papillae in the shape of an inverted V at the back of the tongue. Each of these papillae is surrounded by groove and contains about 250 taste buds. Each taste bud states cell are elongated cells with hair-like processes called microvilli at the tips that extend into the taste buds pore. As you can see in the figure, pores in the tongue allows testins to enter taste pores in the tongue. The food molecules or the testins are dissolved in saliva and they bind with the stimulate with and they bind with this and they bind with and stimulate the receptors on the microvilli. The receptors for testins are located across the outer portion and front of the tongue, outside of the middle area where the filiform papillae are most prominent. Now, lastly but not the least, let's have a look on the effect of age on senses. Both tasting abilities and sense of smell change with age. In humans, the senses decline dramatically by age 50 and continue to decline. A child may find a food to be too spicy, whereas an elderly person may find the same food to be bland and unappetizing. Hope you have enjoyed this session. If you really liked the video, kindly hit the thumbs up icon below and share the video as much as possible. Once again, thank you so much for your love and support. Have a nice day ahead.